Hello, madam. I'm HEERANYAA Nadaraja and I'm from DBT 4A. Today, I would like to perform on the amplification of DNA through the polymerase chain reaction. Before entering to this performance, I would like to give a short intro about polymerase chain reaction. What is polymerase chain reaction? Polymerase chain reaction, also called as PCR, is a fast and inexpensive technique used to amplify copies of small segments of DNA. Because significant amount of a sample of DNA are necessary for molecular and genetic analysis, studies of isolated pieces of DNA are nearly impossible without PCR amplification. PCR is one of the most important scientific advances in molecular biology. Kari V. Mullis is the creator of the PCR who was awarded the Nobel Prize for Chemistry in 1993. PCR is also valuable in a number of laboratory and clinical techniques including fingerprinting, detecting bacteria or viruses, or some particular for AIDS and diagnosis of genetic disorders. Meanwhile, PCR can achieve more sensitive detection and higher level of amplification of specific sequence in less time than previously used method. Let's launch the interactive. Here we have the introduction for our performance on the PCR. Here we have the basic steps for this performance. First, we will prepare a sample from a patient and isolate whole bacterial DNA. Make many copies of the desired piece of the DNA using the PCR. Then sequence the DNA, analyze the sequence and identify the bacteria. So probably this is the importance of the PCR. The piece of DNA will be used for identifying the bacteria in the region that codes for a small subunit of the ribosome RNA. Then we will refer to this piece as the 16S rDNA. Different bacterial species have unique 16S rDNA sequences. The identification relies on matching the sequence from your sample against a database of all known 16S rDNA sequences. So here we have the objectives of this performance. We will get to know that the kind of patient's sample are used for the purpose of identifying possible pathogens. Then we will know the importance of the PCR, how does it work, how it is useful. Then we will get to know to separate the described DNA from all others. Then we will know an automatic DNA sequencer work, and then we will get to know the possibilities of the usage of a DNA sequences to identify the bacteria. Now, here we have the virtual lab performance. So let's start to click it to enter. Here we go. Here we have 6 parts, which is sample preparation, PCR amplification, PCR purification, sequencing preparation, DNA sequencing, and sequencing analysis. This is our first part, sample preparation. So here we have the virtual bacterial ID lab. To begin, let's put a pair of rubber gloves by clicking the glove on the box. So before entering or performing on this simulation, we have to wear gloves, and then we should click on the wire loop to continue to prepare the sample. So this is our bacteria, and this is the small colonies on the agar plate. So we have to sterilize the loop until it gets red, and then we should take it out one clone from this agar plate, and then we have to click on the microcentrifuge tube to transfer the bacterial clone from the wire loop. After successfully entering the clone into the microcentrifuge, let's click on the yellow micropipette for us to add the digestive buffer to the microcentrifuge tube. This buffer probably will, we can say like it can split, or it will smash the DNA into the bacteria, and then you have to click it on the microcentrifuge for several hours for us to make the buffer to react towards the bacterial clone. The DNA inside the bacterial clone can be smashed and can be lysis. Yeah, lysis. So after few hours, the DNA into the bacteria clone that we placed into the microcentrifuge tube will be lysis. And then we're going to click on the microcentrifuge tube to inactivate the digestive enzyme. Throughout this process also the lysis reaction will occur, and then we're going to click it on the microcentrifuge for the process, which is we're going to centrifuge it, close it the tube, make sure the centrifuge should be in balance, and then we're going to click it on the microcentrifuge. We are taking the supernatant from this microcentrifuge and we are transferring into the PCR tubes. There are 3 samples, so we are transferring it into the tubes, and then here we go. We're having part 2. Part 2 is PCR amplification. First step, we will add the master mix. This is the master mix. The master mix will be added into the microcentrifuge, and then we will run the PCR. Here we have the PCR machine. So we will run the PCR machine according to the temperature 95 degrees Celsius for 10 minutes. So let's start. Now we are adding the master mix to the sample, to the PCR tube. And then now we are adding the positive control PCR reaction tubes into the adding the control DNA into the PCR tubes, and then we're going to we're adding water to the negative control PCR tubes, and then let's click on the PCR machine for the further process. Now we are entering into the part 3, which is PCR product purification. Here we contain many copies of the 16S rDNA, each about 1,500 base pairs long. At this time, we have to run a gel to confirm that the PCR reaction will be work. The gel should contain 3 lands, one for the negative control, one for the positive control, and another one is for our sample. If we're confident that the PCR will be work, we can proceed with to purify the PCR product. Running a gel is actually one of the method of purification. Once the PCR product is in the gel, we can cut the band corresponding to the PCR product and isolate the DNA from the gel. Nowadays, we can buy a compact microfilters to filter the DNA from the PCR tube without running the gel. We can use such microcentrator columns in our procedure. Here we have 6 procedures for us to perform in this part 3. First, we have to insert the microconcentrator column of appropriate size into a collection tube. So here we go, we're having the buffer. So add about 400 microliters of buffer to the column. Here we are having 400 microliters. We just added the 400 microliters of the buffer to the column, and then we are going to add the entire PCR content to the column. Here we are having the PCR product, so we are entering it into the column, and then we are going to spin the column at 3000 rpm in a fixed angle centrifuge for 15 minutes. Here we are closing it, and now we are waiting for 15 minutes. The PCR product should be trapped into the column, while the collection tube should contain all the primers, nucleotide, and other small components that we no longer needed. So we are going to remove the collection tube and discuss it. Invert the column and attach to a new collection tube. So here we are having a new collection tube. So we are attaching it into the new collection tube. Now we are adding about 50 microliters of buffer to the inverted column. And then now we are going to throwing the old column into the trash box, and then now we are going to spin the inverted column at 3000 rpm for 2 minutes to collect the sample in the collection tube. Yeah, you are having 2 minutes. The final collection tube should now be many pieces of 1500 base pair long of 16 srDNA with a small amount of longer DNA strands. Now we are going for the part 4, which is sequencing preparation. So here the DNA sample has been purified. So our PCR tube will be contain almost nothing, and but the copies of the 16 srDNA will be there. So we can prepare the sample for automatic sequencing. DNA sequencing technology is another of the molecular biology that has been impressive amount of refinement. So now we are going to dilute the PCR product to increase the volume. So let's click on the blue cap distilled water to continue it. Now we're adding the distilled water into the purified PCR product. So now we are adding the purified PCR product to the each tubes. for the green tubes. And now we are adding for the blue tubes also. Then now we are going to place it into the PCR machine. After the running the PCR amplification, so here we can see the process of it, the fragments of the DNA. So here we can see the length of the fragments of the DNA. So after we finish, take it down. Let's enter to part 5, which is DNA sequencing. We are having currently we are having 12 tubes that contain the final PCR product, which is a mixture of the DNA pieces of variable length. All these DNA pieces in each tube will be start with the same primer but end with a different nucleotide tagged with the fluorescent marker. Therefore, we are using an automatic sequencer that performs the gel electrophoresis on the DNA in each tube. Gel electrophoresis is a method that used to separate the biomolecules based on the differences in size. The sequencer will be used in this lab has a thin capillary tube attached at one end to a syringe machine that contains a buffer solution. Since the molecules are negatively charged, they start to move through the tube toward the positively charged syringe end, with the smaller pieces moving faster than the larger ones. So the smaller DNA fragment will move fast than the larger DNA fragment. At the end of this part, the sequencer automatically flushes out the buffer from the tube, moves the tray, and runs the electrophoresis again, repeating the program until all 12 tubes have been examined. The results will be set of sequences are collected by the computer program to build the complete sequence of the 16SR RNA gene. So let's start by clicking the orange tray. Here we are having the DNA sequencing machine. So we are placing the orange tray into the DNA sequencing machine. So this is known as the inner gray door. So we are clicking it into the inner gray door. Here we can see the curves, various nucleotides representing various color of curves. So there are different colors representing different nucleotides. By presenting the different colors for different nucleotides, it's easy for us to view and to identify the nucleotide. Next. Now we are entering into the part 6, which is DNA sequence analysis. So just now we did the bacteria identification process for the 6 samples. So here we go. Now we are having a sample A, which is fluid from the lymph node. So let's start to identify the bacteria by clicking on the computer screen. After clicking the each one of the options that we found from this box, here we found the actual bacteria from the each sample. So first, sample A is the fluid from the lymph node, and the identification bacteria is Bordetella hanseatic. So this sample was obtained from the tissue and fluid aspirated from the lymph node of a patient who has a fever. And then sample B is the stool sample, and the identification bacteria is Escherichia coli, which is known as E. coli. So this sample was from the stool of the patient with a normal temperature complaining of the severe abdominal cramps and watery diarrhea. And sample C is the urine sample, and the identification of the bacteria is the Pseudomonas aeruginosa. So this sample is the we obtained from the urine of a patient who complaining of having a pain during urination and discharge from the urethra. And next, here we are having sample D, which is a blood sample, with the bacteria of Salmonella typhimurium. So this sample was obtained from the blood of a patient who complaining, who having a fever, headache, joint pains, constipation, and so on. Furthermore, sample E is a sputum sample, and the identification of the bacteria in this sample is Yersinia pestis. So this sputum of a patient complaining of chills, high fever, and a cough with elaborate breathing and sputum with flecks of blood. Finally, here we are having sample F, which is a stool sample, which contain the bacteria which is known as Yersinia enterocolita. So this sample was obtained from the stool of a child who has a fever, abdominal pain, and the bloody diarrhea. So here we go. For each sample, we has identified the suitable bacteria inside the sample. So that's all from me. Thank you, madam.